Hey, Michelle.
City rock, get on your feet. Baby, cellular construction has DNA instructions for base simple code direction, peptide production, quick stainless deduction, started from induction, a simple path to follow. DNA to RNA, to protein, the settled on the song. DNA to RNA, to protein, it's almost never wrong. Hydrogen bonds, C to G and T to A, double-stranded DNA, backbone sugar phosphate, nucleotide base, make it chuck but don't hate, information they relate, in control of your traits, replication, it makes the DNA copy semi-conservative, and it's not that sloppy, but just I got DNA polymerase.
Oh my god, I cut myself.
Here, let me see.
Don't touch it.
Oh,
I'm gonna die.
Michelle, calm down.
bleeding.
Oh my gosh, Michelle.
Oh my
Seriously,
God.
calm down. You have 3 lines of
Just cut my
defense.
hands off. What?
Yeah, 3 lines of defense. You have the first line of defense, which is your skin mucous
But
membrane.
it just cut through my skin. It's bleeding.
Michelle,
Did you see that?
then you have your second line of defense, which is your white blood cells, your defense proteins, the inflammatory response, and fever.
Fever?
Okay, well then you have your third line of defense, which is your T cells, B cells, and antibodies. So you have nothing to worry about.
Really?
Yeah.
It's awesome. So I can like cut myself all I want and I'll still live or something. Like,
Well, you'll live, but let's not go crazy now, okay? Yeah.
Michelle, you're doing homework on a Friday night?
Well, sort of. It's for biotech. We actually have to make this little YouTube video on what we learned in class, and I'm doing a skit. Thank God. The other option was the final, and that does not sound too fun at all.
Biotechnology? What is that? Is that like engineering?
Sort of, not really. It's kind of like, well, the technical definition is the study and manipulation of living things or their components like tissues and cells. But the thing is, biotech is a huge field of study, so it's kind of hard to pinpoint exactly what they do, but it involves working with a lot of DNA and molecules and proteins and different technology.
What kind of technology?
Well, like recombinant DNA technology, for instance. A lot of modern pharmaceutical drugs like insulin and such wouldn't exist without this technology. So basically, the purpose of this is to bring together genetic material from multiple sources, creating sequences that would not be found in biological organisms naturally. And this is possible because DNA molecules from all organisms share the same chemical structure, and they're only different in their nucleotide sequencing. So when DNA from a foreign source is linked to a host sequences that can drive DNA replication, it's then introduced into a host organism and the foreign DNA is replicated along with the host DNA.
I don't think I really get it. I'm a visual learner.
Here, I'll show you on my whiteboard. I think it's easier if I show you a specific example with insulin. So, basically, you have human cells, which is the source of the insulin gene, right? And then obviously they have DNA that contains the wanted DNA. And then on the other hand, you have a spliced plasmid vector, and these right here are sticky ends, right here. And then restriction enzymes cut a portion of the wanted DNA, and then it puts it into the plasmid right here. Here, I'll draw a different colored pen. With DNA ligase. So now you have this plasmid, and then with both species of DNA, and then this is finally inserted into an E. coli cell. It's usually, you know, an E. coli cell, but it can really be any cell that the scientists prefer. So this is all the chromosomes, and then this is the plasmid that we created. And then finally, this cell, you know, recombines and it reproduces, and then finally it creates the recombinant human insulin that's made in bacteria. So that's where you get your drug, and that's what, you know, the epitome of our DNA technology is.
Oh, I see. But so does that mean that you could make dogs glow and have plants resist pests?
Sort of, and that's where GMOs come in.
Hey Michelle, what are you doing?
Oh, hey Kara. I'm eating a grapple.
A grapple? What's that?
Yep, it tastes like a grape, but it smells like an apple, but it's actually, I mean, look at this. It looks like a strawberry, you know?
What? That's impossible.
Yeah, a genetically modified organism is an organism whose genetic material has been altered using, you know, engineering techniques like rDNA technology, which uses DNA molecules from different sources, which is combined into one molecule to create a new set of genes. And this DNA is then transferred into an organism, giving it modified or novel genes with desirable traits. And in this case, the genes of a grape and an apple were introduced into the genes of a strawberry, hence the grapple, which, well, you know, it just looks like a strawberry right now, but yeah.
That's really interesting.
It is. Can I have my grapple back now?
Maybe later. You want to go to a bonfire on Saturday?
Oh man, it's this Saturday?
Yeah.
Oh yeah, I can't because I have to drop my dad at the airport. He's going to Korea this Saturday for, you know, his job and stuff, and I have to drop him off.
I thought your dad worked at Sequina in San Diego.
Oh, he did. Girl, where have you been? Haven't you heard about the Sequinum scandal that happened in 2009?
Oh, I heard about it, but I don't really know all the details.
Yeah, it's kind of personal, but I think it's important that you know because it really shows how important bioethics is. Here, I'll show you on the computer. There's actually lots of news articles on it. It was really big, like all over the news. And you know, before I tell you about it, you know that it takes a product maybe decades to be successfully launched into the market, and not everything is a guarantee either. You know, it has to pass, you know, numerous clinical trials, and to make it worse, keeping all this up is really expensive, so it takes hundreds of millions, and obviously most companies don't have that much money, so they have to get grants or funding from the NIH, or, you know, get money from private investors. And the thing is, if, you know, tests show that a particular drug or product is ineffective or show faults, the company loses virtually everything. And what's more, in public companies like Sequinum, millions of dollars also depend on the stocks in the company too. And, well, with Sequinum, let's read the news article here. I pulled it up right now. Right here, you can see it, Sequinum scandal. Key figure in Sequinum scandal is dead. And the person that was behind this whole scandal deal thing actually died 2 months ago from cancer. Alright, it says, the former head of research at the genetic testing company pleaded guilty in June 2010 to lying to investors and stock analysts about clinical trial results for the company's experimental genetic tests of blood from pregnant women to determine if their unborn babies have Down syndrome. She was facing up to 25 years in prison and more than $250,000 in fines. Okay, so that's one part of the article, and then a little bit more down. It says, in early 2009, Sequinum was riding high on positive clinical trial results for a Sensi gene, which was scheduled to go on sale on June 2009. But suddenly, April, the company said that the launch would be delayed because employees had mishandled supporting data. And it set the company's stock stumbling at 82%. And here it says, Betty Dragon and the company's chief executive officer, Harry Stiley, were fired, and 2 other senior executives resigned. But Betty Dragon was the only one to be charged with crimes tied to the scandal. And this really shows that it's a life and death matter. Like, companies take this really seriously. You know, bioethics, that kind of stuff that we learned, you know, it's not something to be taken lightly, and especially, you know, if it has to deal with drugs and biotech, it's important that we use honesty at all times, because it involves so many people's jobs. I mean, like, you know, even though Betty Dragon was the only one that was directly involved in the scandal, who schemed this whole thing up, my dad and several other people and executives had to resign because they worked in the same department as she did. So it kind of shows how seriously the real world takes this kind of bioethics deal.
Wow, I guess what Ms. Dixon taught us about bioethics really does happen in the real life.
Sure does.